All right, so we asked the question, how is it possible that if two individuals are phenotypically normal, how is it possible they can still have an offspring with cystic fibrosis? So we actually use Punnett squares to determine the probabilities that a couple are going to have, in this case, a child with cystic fibrosis. So when it comes to Punnett squares, we use the genotype of the parents. So in this case, we have a gentleman who's heterozygous, so which means he has a dominant allele and a recessive allele, and then we have a lady who has a dominant allele and a recessive allele. So we use a Punnett squares to determine the probabilities of a couple having offspring with a specific trait. In this case, we're focusing on cystic fibrosis. So let's figure out, how do we do Punnett squares? All right, so the way to do a Punnett square is that we have a big box in the middle, and inside the big box, I have 4 small boxes. So on the top, I have the genotype of the dad. In this case, we're talking an individual that is heterozygous, so that's represented by an upper letter A and a lowercase a. And in this case, I'm putting the lowercase a of a different color. In this particular case, is red, to represent that that's where the mutation is. On the left side, I also have the genotypes of the mom. In this particular case, we're also talking about a heterozygous individual, which is represented by a upper letter A and a lowercase letter a. And once again, I'm using a different color, in this case red, to represent that the mutation is in the lowercase a, which is basically the recessive allele. And so the way basically to go about identifying the probabilities is that you basically multiply the genotypes of the mom with the genotypes of the dad. So in this particular case, I'm using arrows to represent, if you will, the multiplication. So I'm multiplying the dominant allele of the mom with the dominant allele of the dad, and then I'm gonna multiply the dominant allele of the mom with the recessive allele of the dad, and then I'm gonna move towards the recessive allele of the mom, and I'm gonna apply that with the dominant allele of the dad, and finally the recessive allele of the mom with the recessive allele of the dad. So let's see how what we get. Okay, so basically this is what we get. So on the first box, we actually have an individual with 2 dominant alleles. So this individual is going to have a normal phenotype. And then if we look at the other boxes, 2 of the boxes we see an individual that is heterozygous, which basically means that we see a dominant allele and also a recessive allele. So in this particular case, both of those individuals are going to be what we call carriers, which means that they're going to have a normal phenotype, but they can still pass on that mutation to their offspring. Okay, and finally, we have an individual with 2 recessive alleles. So in this particular case, he has both mutations in those alleles. So that means that this is the individual that's going to have cystic fibrosis. Okay, so the way to determine the probabilities is that you basically count how many of the individuals have both the recessive alleles. You divide that by the total boxes, if you will. So in this case, it's 4, and so that's 1 in 4, or a 25% chance that this couple is going to have an individual with cystic fibrosis. So what about if the mutation is in the dominant allele? So what are the probabilities that a couple is going to have an offspring with that specific trait? Okay, because this is a mutation that is happening in the dominant allele, you only need one of those alleles to be mutated in order for that specific offspring to actually have this specific trait. Okay, so here it's a couple, and both of them are actually heterozygous. So the genotype of the dad is represented on top, and what I have is basically the dominant allele is represented by an uppercase letter A, and in this case, that uppercase letter A has a different color. In this case, it's red, basically to show you that that's where the mutation is. And I also have a lowercase letter a. Okay. Now, when it comes to the genotype of the mom, again, because she's also heterozygous, so she's represented by 2 letters. So I have an uppercase letter A. In this case, the uppercase letter is of a different color. In this case, it's red to represent that's where the mutation is. And then also a lowercase letter a. And we basically multiplied the the genotypes of the mom and the dad. So in this case, I'm going to multiply the uppercase letter A of the mom with the uppercase letter of the dad, and then the uppercase letter of the mom with the lowercase letter of the dad. And then I'm going to move towards the right, so I'm going to multiply the lowercase letter of the mom with the uppercase letter of the dad, and finally the lowercase letter of the mom with the lowercase letter of dad. So let's see what happens. All right. So once we do this, then we actually can see the Punnett squares. So one of the things that we can see is that 3 of those individuals are actually going to have that specific trait. So the reason for that is because, remember, because this is caused by a dominant allele. So as long as one of the dominant alleles has that specific mutation, then that individual is going to have that trait. Okay, so in this case, when we count the number of offsprings that could have that mutation, it's 1, 2, 3 divided by the total. So we have 4 squares. So 3 divided by 4, that's a total of 75 or 75%, if you will, of the individuals are going to have this specific trait. So in this particular example, having the recessive allele is actually beneficial, if you will, if we were talking about a disease. Okay, but remember, you can use Punnett squares to determine traits, and a trait can be, for example, having freckles, having eye color, and so on and so forth. All right, but in real life, most of diseases and also the traits are actually caused by multiple genes. So you also have to take into account the effect of the second gene. So in this particular case, since we're talking about cystic fibrosis, you also have to take into account what is the effect of the transforming growth factor beta 1. So this is a gene that is important for cell differentiation, cell proliferation, cell growth, and also important for apoptosis. So if you guys remember, apoptosis is cell death. So whenever your body cannot fix a specific mutation, then that cell takes one for the team and you basically commit cell death. Okay, so all right, so let's look at a Punnett square that takes into account the effect of the second gene. All right, so in this case, we have a couple that are heterozygous for both of these genes. Okay, so in this case, the alleles for the CFTR gene are represented by letters A, and the alleles for the TGFB1 gene are represented by letters D's. Okay, so the first thing that we need to do is to determine what are the gametes that can be produced, in this case, by the gentleman. All right. So the first gamete that he can produce is basically a gamete that is dominant for both of these alleles. Okay. And so the second gamete that he can produce is a gamete which is dominant for the CFTR gene and is recessive for the TGFB1 gene. And the third gamete that he can produce is a gamete that actually has the mutation in the CFTR gene and is dominant for the TGFB1 gene. And the last gamete that he can produce is a gamete that is actually recessive for both of these alleles. So it actually has a mutation in the CFTR gene. And the same thing happens when it comes to the female as well. Because she's also heterozygous, those are the only same gametes that she can actually produce. So once we do the multiplication, so what we see is that 4 of the offsprings are actually going to have cystic fibrosis. And if you guys remember, cystic fibrosis is caused by having mutations in the recessive alleles. Okay, so in this case, it's basically a total of 4. So 4 offsprings out of a total of 16 are going to have cystic fibrosis. But because we're also taking into account the effect of the second gene, even though these offspring are going to have cystic fibrosis, they're going to experience different symptoms. So for example, some of them are going to experience a milder symptoms when it comes to cystic fibrosis. And the last thing that I would like to say is that about 20 years ago, the average lifespan for an individual with cystic fibrosis was close to 30 years. So they died basically fairly young. But because scientists are working very hard on better treatments, on better cures, now actually the lifespan of an individual with cystic fibrosis has increased by 10 years. However, that this is not enough. So there's still, people who are working very hard in hopes of actually finding a cure for cystic fibrosis. So hopefully this was helpful. If you guys have any questions when it comes to this lecture, make sure you email your professor. We are always happy to receive emails. We're always happy to answer your questions. So thank you and stay safe.